All right, welcome back. So we are going to continue discussing digestive system. So this is the second part of your lecture. Let's start it. So we are going to talk about some of the glands which are associated with the digestive system, or we can say is accessory organs present in the digestive system, which is the liver. So first we will talk about the liver, its general anatomy and microscopic anatomy. We'll also talk about the gallbladder, the bile, the pancreas, and then we'll talk about the large intestine, and then a little bit about the chemical digestion. Okay, so the liver. So the liver is the largest gland in the body. And where is it present? It is present in your upper right quadrant, more towards the right side, and it has four lobes. One of the largest lobe is the right lobe, and then we have a left lobe. And you have to look at it from posterior side or the dorsal side to find the third and the fourth lobe. So the third lobe is caudate lobe, and then the fourth lobe is quadrate lobe, which is down here. Okay. Uh, the four lobes, the first lobe, which is the right and the left lobe, they are connected, or you can say that it is actually separated from one another by a connective tissue ligament, what is known as a falciform ligament. And the falciform ligament at the base of it ends into or hangs into what is we call as round ligament, or also known as ligamentum teres. Okay. When you look at the back side of the liver, you see a depression in it, a huge depression here. And this depression actually is where we have the sulcus. That means the inferior vena cava goes behind it. And it also have a porta hepatis, means this is the portal from where the different blood vessels and lymph vessels go inside it. So the porta hepatis is this region in the back. And you also see gallbladder here in this picture also, and the green one in this picture. Okay. In the porta hepatis, you have a hepatic portal vein, which brings in the nutrient-rich blood from the intestines to the liver. And then we have the hepatic artery, which is the proper one. And then we also have the lymphatic vessel, which is the common hepatic duct present. Okay. So, and this is the in hepatic veins, and then in also we have where the sulcus is present for the vena cava. Okay, so they come out. So these are the important things you can see at the surface for the liver. Okay, so this is macro anatomy of the liver. So other thing which we see is the gallbladder. Okay, so the gallbladder, and it is connected to the liver through certain ducts, and liver synthesizes the bile which is stored in the gallbladder. Okay, so the bile ducts which are there, one comes from the right lobe and other comes from the left lobe. So these are known as right and left hepatic ducts of the liver. They merge together to give you what is known as the common hepatic duct. And then part of this common hepatic duct, there's a duct coming from the gallbladder which is known as cystic duct. They combine together to give you a bile duct. So bile duct then travels inferiorly, and it comes and meets the main pancreatic duct, and they form a small area here, what is known as the hepatopancreatic ampulla. And then there is a sphincter present right here, and that controls the release of the bile and the pancreatic juices based upon when the food enters into the duodenum. Okay, and as mentioned earlier, also the accessory pancreatic duct also opens up into the duodenum at another place, but the main pancreatic duct forms or combines with that of the bile duct. And the opening of both the bile duct and the main pancreatic duct is in a small raised area inside the duodenum, which is known as the major duodenal papilla. Okay, what's the function of the bile? The function of the bile and also the liver. So the liver synthesizes lots and lots of plasma proteins. It also helps us to phagocytize worn out RBCs and also store nutrients, do gluconeogenesis, glycogenesis, etc. It also breaks down toxins and also produces the bile. The bile helps us to digest the fats. Okay. Okay, so let's look at the microscopic anatomy of the liver. So the liver is made up of small uh, small little sesame seed type of lobes, and those lobes can be considered as if you have a book and you fold the pages of the book into so hold on, I am just gonna grab one. So if this is the book and you fold the pages of the books like this into and put them like this together into lobes. Okay, so they kind of fold like this, and this lobes are like this. So if you fold the whole book like this, all pages like this, and they fold them like this, and then the central region will be a central canal, and these lobes are made up of like a hexagonal structures. Okay, and they contain these plates of cells which make the liver, which is known as hepatocytes. So the hepatocytes actually filter and process the nutrient rich blood. Okay, and at the end of this hexagonal structure, at every sixth corner, there is a small three vessels together, what we call as the portal triad. Okay, so what is in the portal triad? You have a hepatic artery branch which supplies the blood, which is the red color. Then you also have the branch of a hepatic portal vein, which brings in the nutrient rich blood from the intestine, digestive tract. And then we also have a bile duct present, which is the green color in here shown. Okay, so do not confuse this bile duct with that of a lymphatic vessel. Okay, and these foldings of the folding is like bricks of or walls of these hepatocytes present there. Okay, in between these hepatocytes, the cells are connected through small canals, and these canals are known as bile canaliculi. So the blood actually comes from the portal veins, and it actually moves inwards like this, and as it goes, same way the blood from the artery, the portal arteriole also goes in, and they gets mixed up. So the nutrients actually are absorbed by these hepatocytes which are present there, and where this blood mixes up is called the sinusoids, and eventually the blood drains inwards like this direction into the central vein, and then from the central vein it all gets into the interlobular veins, and then from there it goes to the hepatic veins, and then eventually to the vena cava. Okay, the bile, in the other hand, is moved through these canaliculi towards the bile duct. Okay, so the bile travels in the opposite direction of the blood. That's the important fact to know and remember. So let's look at this picture. This is a little bit better picture to give you some idea about. So this is the central vein here. This is the hepatic triad, portal triad. So you have a hepatic artery here, and then you have the portal vein, and then you have the bile ones, and these are the layers of the hepatocytes. So you can see this, these are the sinusoids. This area is the sinusoids present, where the blood gets mixed up, and then changes into the venous blood and goes in there. And the hepatocytes produce the bile, and then the bile is collected in between what we call as this canaliculi. So there's another slit like between the two rows of the hepatocytes, and then it moves towards this bile duct. Okay. Liver also have some free cells which are known as macrophagic cells, and these are fixed cells which never leaves the liver. We call them Kupffer cells. So these Kupffer cells are white blood cells, and they are always there, and what these cells do is they remove any dead cells or if there is any bacteria present in there. Okay. Uh, Looking at the hepatocytes, so hepatocytes are actually modified cells, kind of epithelium looking cells, but they have lots and lots, large amount of rough as well as the smooth endoplasmic reticulum, plus lots of Golgi apparatus, peroxisomes and mitochondria, etc. If you look at this, this is one lobule. The central vein is here, and if you look into it's little bit magnified, you can see that in here you can see these sinusoids present, and here's the sinusoid little bit larger, and then you can see the hepatocytes. These are the large hepatocytes with the nucleus, and they have lots and lots of vesicles. Why do they have lots and lots of vesicles, rough and smooth ER or Golgi apparatus? Is because of the because they do a lot of anabolic reactions, means they synthesize things. One of them is the bile salts are being made. Okay, so hepatocytes typically produce almost 1 liter of bile per day, and they also process a lot of bloodborne you know nutrients. For example, RBCs. Also, they also produce glycogen from extra glucose we have. They produce a lot of plasma proteins. They also produce and detoxify a lot of drugs or any kind of poisons the body is exposed to. They also convert the ammonia which is produced by the protein digestion into urea, and also do store fat soluble vitamins. So liver hepatocytes are very very busy. So actually just looking at the liver pathology, you can actually check if a patient who has died, whether they had been on any drugs etc or not. If somebody has been taking a lot of drugs or has been exposed to a lot of chemicals, their hepatocytes have lots and lots of smooth endoplasmic reticulum. So what is bile? Bile is a yellow-green solution. It's a thick solution, and it is alkaline pH, and it contains many bile salts. It also contains bilirubin. The bile salts actually are obtained from the cholesterol, so they also contain cholesterol also. And then they have a lot of phospholipids as well as other salts. So there are two types of bile salts. We have, one is the primary bile salts, and then we have the secondary bile salts. Okay, your book doesn't mention them, so you do not need to worry about it, but your book does mention that bile salts are released into the duodenum, where they actually help to dissolve the fats into by emulsification process, and then from emulsification process the lipids are absorbed by the intestine, and then the bile is actually recycled too. Okay, there is some of it is lost in the fecal process, but not much. So it is actually recycled in the portal system, so which is the enterohepatic circulation. So it is reabsorbed in the ileum and then through the hepatic portal vein and through the liver. So that's how they actually absorb it. So how does the Okay, let's talk about how does the bile salts help in fat emulsification. So all of us have used, detergents when we do our dishes, and if there is oil, cooking oil present, we put some detergent on it. What happens to the oil? It just breaks down into small little droplets. That's called process is known as the emulsification process. So almost same thing bile salts do in our body. So if you have a large fat globule, it will be broken down into smaller fat droplets. So the bile salts bind to this small fat droplets, and then the pancreatic enzymes can work on them, and they can make uh, free fatty acids and the monoglycerides, which is a glyceride molecule attached to one fatty acid. So then they also bind to more bile salts, and then they form what we call as micelles. Okay, so these micelles then can be carried, and they can be carried inside, they can be absorbed inside the intestinal mucosa. Okay, so once they are present in and they are taken into the epithelial cells, there they can be recombined with other proteins, so that they form what we call as chylomicrons. And then the chylomicrons are excluded out of the epithelium cells, and then they are absorbed by the lymphatic vessels, and that's the lacteals. And then it is carried away from the intestine by the lymph. Okay. So let's talk a little bit about gallbladder. So a gallbladder is a tiny little pouch or a sac which is thin walled, and it is muscular sac which is present on the ventral surface of the liver. Major function of gallbladder is to store as well as concentrate the bile by absorbing water and ions from it. So sometimes the gallbladder, it has little bit foldings inside it, which are known as the honeycomb folds, which actually helps it to expand as it gets fed. Okay, it does have a muscular layers in its body. So when they contract, that helps the bile to move through the cystic duct, and then goes and combines with the common hepatic duct and create what we call as the bile duct. Okay. Okay, so let's discuss a little bit about some of the homeostatic imbalances of the liver. So hepatitis, you probably have heard, and there are a number of different types of viruses which can create hepatitis. We have Hep A, Hep B, Hep C, and even Hep D, etc. These are viral infections which can change the liver cells, and they affect the livers. Okay. You can also have hepatitis from wild mushroom poisoning, etc. Liver can also become fatty liver when it has too much fat deposited in it. The color, so this is the normal color of the healthy liver, which is more red, and it's quite soft. When it becomes fatty liver, it becomes kind of harder and fibrous, and then it can actually increase what we call as portal hypertension. And then liver can change into fibroid, or liver can also have cirrhosis. Cirrhosis is when the function is almost gone, and liver cirrhosis can happen because of alcoholism, chronic inflammation from chronic hepatitis, etc. And liver can regenerate itself. So liver transplant is done, and actually one healthy liver can be given to 3 patients depending upon what's the condition of the donor liver, and then it can generate itself to full size within a year or so. Okay. Another homeostatic imbalance which is very common is that of the gallstones, or what we call as biliary calculi. Okay, this is if you have too much thick bile, and it has lots and lots of cholesterol and very few bile salts in it. So these stones can be formed, and they can actually obstruct the cystic duct. They can obstruct the cystic duct, or they can obstruct the common bile duct, or they can also obstruct you know um, the bile duct, or at the end when it combines with the pancreatic duct. So that can be painful when the gallbladder try to contract and to have the bile released. Okay, so what happens is as the bile is uh, then the bile is not released, so this can cause jaundice accumulation. Now there's two types of jaundices. You can have an obstructive jaundice, which is the blockage and can cause the bile pigment salts and pigments to build up in the blood, and then you have a yellow color skin. Okay, so typically jaundice can also be caused by when the liver doesn't function very well, because then the liver is not removing the bile salts or broken down hemoglobin, etc., from the blood. Typical treatment for gallstone treatment is you can have crystal dissolving drugs, you can have lithotripsy, which is the sound wave treatments. You can also do laser vaporization, try to do that, or you can actually have surgery to remove the gallbladder. The second important uh, gland in the digestive system is the pancreas. Okay, so pancreas actually sits right snuggled in between the duodenum here. Okay, so the pancreas, the body of the pancreas is there, this side is the tail of the pancreas, and this is the body, or you can say base of the pancreas is there. Okay, pancreas is a retroperitoneal. Okay, and it just fits into the C shape and duodenum there. Pancreas is both endocrine and exocrine gland. Okay, in an endocrine gland, it secretes two hormones. One is insulin and the other is glucagon. Their actions are in opposite to one another. So they both regulate the blood glucose levels. Insulin decreases the blood glucose level. Glucagon increases the blood glucose level. The exocrine function of the pancreas is that it secretes what is known as the pancreatic juice, and this is done by epithelium cells which look like glands, and there is a group of cells together with a duct in the middle, and that is known as an acini. Okay, so this acini, for example, here is shown. So you have many acini present in the pancreas, and the lumen is there. So all the acinar cells, they secrete uh, their enzymes, etc., which are always in inactive form. We call them zymogen forms. They release them into here, and then these small ducts combine together to become bigger ducts, and finally in the pancreas we have the one main duct, what is known as the main pancreatic duct, and then there is also a smaller another accessory pancreatic duct, and both of them actually opens up into the duodenum, but the main pancreatic duct combines with the bile duct, and they have sphincters, and then those sphincters are there, and then they open up into an ampulla, which is a raised area inside the duodenum. The accessory pancreatic duct also opens up into the duodenum, but this one does not open up into any raised ampulla present in the middle. Okay, and here are few of these cells present, and you can see the duct is there. So these are the acini cells present here. Insulin is released by the beta cells present. So the pancreatic juice is anywhere from 1.2 to 1.5 ml per day, and it typically contains the water, the salt, the electrolytes. The bicarbonate ion is required so that it can neutralize the chyme, which is highly acidic, and then we have many enzymes which are released. Okay, so those enzymes are amylases, pancreatic amylases, which work on carbohydrates. Lipases are there, and then nucleases are there, which are all secreted in the active form, but they may require ions or other or bile for their optimal activity. Proteases also are secreted, but the proteases are secreted in the inactive form. So for example, trypsinogen. Trypsinogen is changed into trypsin by membrane-bound cells, which is enteropeptidase. Chymotrypsinogen is released in inactive form, and then the trypsin activates chymotrypsinogen into trypsin. You also have procarboxypeptidase, which is then changes into carboxypeptidase, etc. The Another important thing to remember about is the bile and the pancreatic secretion, which is secreted into the duodenum. And we have already talked about it, that the main pancreatic duct as well as the bile duct, they combine together, and they have sphincters at their ends. Okay, and they open up into the major duodenal papilla, and the sphincter is the one which is the hepatopancreatic sphincter here, is the one which controls the release of the bile and the pancreatic juice. And the accessory pancreatic duct opens up here. What regulates the bile and the pancreatic secretions? Okay, so the pancreatic and the bile secretion and is stimulated by the nerve supplies also, as well as the local hormones. Okay, the hormones which really play important role is the cholecystokinin and secretin. Okay, so these are called enterogen hormones, and they are produced inside the intestines. And so whenever there is a bile secretion is increased by the action of cholecystokinin as well as the secretin. So the cholecystokinin is the red dots, and then the yellow color dots are your secretin. Okay, so both of them are secreted by the enteroendocrine cells. So the CCK is stimulated when the proteins and the fats are present in the chyme, and secretin is released when there is acidic chyme coming into the duodenum. Okay, so once they are secreted, they enter into the blood circulation. Okay, so CCK is going to go and act on the pancreas, and then what it does is, so the pancreas will start secreting its juice. The secretin causes the secretion of bicarbonate ions and in the pancreatic juice. Okay, so the vagus nerve is the nerve which actually can also affect that, and can stimulate your cephalic and gastric phases. Okay, once the bile salts are also released by the cholecystokinin. So the cholecystokinin acts on liver also. So it acts on the liver and enhances the bile salt production. Your secretin does not affect that much of the bile salt formation. Cholecystokinin also works on the gallbladder, and it enhances their contraction. Vagus nerve also can affect the gallbladder's contraction. Okay, CCK also causes the hepatopancreatic sphincter to relax, and then hence increasing release of the pancreatic juice into the duodenum. So those are the ways they can affect. Okay. Now let's look at the digestion inside the small intestine. Okay, so once the chyme comes out into the duodenum, so what do we have in the stomach? We have partially digested carbohydrates, and proteins are digested because of the acid, and we have lots and lots of undigested fats. So those things move into the small intestine, and the food stays in the small intestine anywhere between 3 to 6 hours. Small intestine will absorb most of the water. All the nutrients are absorbed. Okay, so small intestine and stomach, they do not play any role in ingestion or defecation. Okay, so slowly, as the acidic chyme is delivered and hypertonic chyme is delivered, this in the small intestine we have the delivery of bile, the enzymes, and the bicarbonate ions from the liver and the pancreas, and then we do is mixing of all this stuff. So how does mixing occurs? So in the small intestine, the mixing occurs by segmentation. Okay, so the segmentation is initiated by the pacemaker cells, which are present in the myenteric and also the submucosal nerve plexus. So these are your intrinsic pacemaker cells, and they will move the things slowly forward. What did they do is overall they push everything towards the ileocecal wall, but the contraction occurs at non-adjacent areas. So what it does is, if the contraction occurred here and contraction occurred here, then they actually mix and and then the contraction is going to be at different place. So it just does is it takes a little bit upwards, a little bit downwards, and then it just keeps mixing it up. Now the intensity of these is also controlled by long and short reflexes, which we talked about in the other lecture. Parasympathetic nervous system actually enhances the motility in the small intestine, and the sympathetic nervous system actually decreases it. Okay, segmentation and peristalsis decreases in a fasting phase. If you do not eat, then the motion actually goes down. The next motion is peristalsis, which is the movement in one direction, and this is done by the adjacent segment contractions. As I mentioned earlier also in the first part, that it is like as if you have a long sock and you put a ball in it and you want to push it to the other end. So it's going to be adjacent areas are contracted. Okay. It also is affected by a hormone, and that hormone is known as the motilin, and this is started in the late intestinal phase, and it can occur every 90 to 120 minutes, which is uh, an hour and a half to 2 hours. And then it's a wave together. In if you look inside here, it's kind of a wave, and it starts from the distal to the previous areas, and all the meal remnants what we cannot digest, the bacteria, the debris, everything in the large intestine is moved like that. Okay, so the things move from the duodenum to the ileum. Approximately takes 2 hours for them to reach into the large intestine. What controls the motility in the small intestine is typically the local enteric neurons. Okay, so that's your myenteric plexus present there. These are the ones which control it, and these are your acetylcholine sensory neurons. So they release acetylcholine, so we call them cholinergic sensory neurons. Okay, so these neurons, when they fire, then they actually enhances the contraction of the circular muscles proximally, and then of the longitudinal muscles distally. So then they force the chyme along the track, and then they also relax the ileocecal sphincter, which admits the chyme into the large intestine. And you also have what we call as gastrial reflex, which can enhance the force of the segmentation in the ileum. And then gastrin also increases the motility of the ileum. Uh, when the ileocecal sphincter or valve closes down, then the chyme exerts a backward pressure, and then and that also prevents its regurgitation into the ileum. And it closes when the backward pressure is there, so the chyme doesn't go back into the ileum. Okay, so now let's talk about large intestine. So large intestine is the start is from this. This is the ileum opening, and then we have the ileocecal wall, and right this portion is known as the cecum. On one end of the cecum is a tiny worm-like little tube-like structure present, and that's called the vermiform appendix. So the appendix plays a very vital role in lymph system or immune system. It houses or holds the gut bacteria. Then We also have ascending colon. So this way is the ascending colon, and then it moves, it makes a right angle at this corner is known as the hepatic flexor because it's towards the liver, or it's also known as right colic flexure. And then you have the transverse colon, and then we have a left colic or splenic flexure present here. And then we have descending colon, okay, present there. And it goes from there downwards, and then eventually it is sigmoidal S colon, which goes backwards, and that's a retroperitoneal ending up into the rectum and then the anal canal. And we have the external and the internal sphincters at the mouth of the anal canal. Okay. You do see the mesenteries, which binds and holds this large intestine in place. Okay, so one of the homeostatic imbalance which is associated with appendix is appendicitis, is when inflammation of appendix occur if the food goes in here and the bacteria is there and they try to break it down, or the bacterial growth becomes very large and then it becomes septic and can be actually life-threatening if the appendix bursts. So the colon, we already talked about the ascending colon, the transverse colon, and the descending colon, and the sigmoidal colon, etc. Know their positions, what they are called on your exam. You can have questions on that. Let's look at the rectum and the anus. So the rectum actually inside has 3 walls. This picture only shows 1 wall. The other one will be on this side, and the another one this. We call them rectal walls. So they stop the feces from being passed whenever the gas is passed. And then lower portion is the anal canal. So in the anal canal we see this structure. It's the last segment of the large intestine, and it opens exteriorly to at the anus, and it has sphincters. It has an internal anal sphincter, and then it has an external anal sphincter. The internal anal sphincter is a smooth muscle cells which is not under your control, but the external anal sphincter is a skeletal muscle, and that is under your voluntary control. This gives you a control to figure out when can you go to use the bathroom. There are these elevated ridge-shaped structures present in the anal canal, what are known as the anal columns. And then outside the anal opening, or the anus, there is these this line is inside. This is known as pectinate line, and then we have these little sulcuses present. We call them as the anal sinuses. So the large intestine has a couple unique features to it. Okay, so one of them is what we call as tinea coli. So tinea coli is actually 3 bands of longitudinal smooth muscle cells in the muscularis region. Okay, so this is the tinea coli, this whole thing going around. And then it has these little beaded structure, which is kind of a pocket-like sac structures. This is known as the haustrum, or plural word is haustra. And then we have these tiny fat-filled, just membranous tiny sacs. These are known as the epiploic appendages. These epiploic appendages, nobody knows what's their function. Okay, so but these are the unique features of the large intestine. Let's look at the microscopic anatomy of the large intestine. So it does have thicker mucosa as compared to the other, and it has lined up the simple columnar epithelium cells, except in the anus region we have the stratified squamous epithelium cells present there so that they can withstand the abrasion. Okay, there are lots and lots of what we call as the crypts, which is this little portion into the lamina propria. So they are present and they contain lots and lots of goblet cells, so they can produce mucin as the food is traveling through the intestine, so it kind of gives it a less friction present. The large intestine does not have circular folds or villi, but and the cells etc. also does not actually produce any kind of enzymes. So there is almost no digestion occurring in the large intestine. It's only the water is being absorbed in the large intestine. So this picture actually gives you very nice comparison between the small and the larger intestine. So we have the semilunar kind of folds, and we have haustra, whereas we have circular folds in the small intestine. We do not have these large villi which is present in the small intestine. Here we do not have. Okay, so we do have some crypts present. They also, small intestine also have crypts present in the lamina propria, and then the number of different types of cells vary between the small intestine and the large intestine. So we do have very absorptive cells present in the small intestine and the large intestine. So does the goblet cell. Enteroendocrine cells are there too, and then we have what are known as stem or progenitor cells are there, but Paneth cells are extra cells in the small intestine. Remember, they produce defense proteins, and large intestine does not have that. Okay. Another thing is there is a lot of superficial venous fluxes present near the anal canal, which can actually form hemorrhoids if they are inflamed. All right, so now let's talk about what is the function of the large intestine. As I mentioned earlier, that there is no chemical digestion in the large intestine which is occurring. It is typically absorption of the vitamins, water, and electrolytes. And typically it propels the fecal material towards the anus. And also there's a lot of bacteria which is colonizing inside the colon. Those bacteria actually help us. They synthesize vitamin B complex and as well as vitamin K. And large intestine is not important for survival, and it can be removed, and you can do either ileostomy or colostomy, or also they can join the anal junction can be joined together if there is any case of colon cancer, etc., or colon obstruction is there. Another important thing which we mentioned as we were talking about the function of the large intestine is the gut bacteria. So the gut flora is extremely important. Okay, it colonizes in the colon, and it enters from the small intestine or from the anus. And as I mentioned, that it can synthesize vitamin B complex, especially K, and the gut flora can also metabolize some of the host-derived molecules such as mucin, heparin, hyaluronic acid, etc. It can ferment undigestible carbohydrates, and it can actually produce acids and gases, which is typically 500 per day. In the gut, in the colon, there is less oxygen, so the environment is actually quite different than what we expect inside our body. So there may be some viruses living in there and some protozoans are there. The normal bacteria is required in our colon, because if it is not there, then the bad bacteria will take over. And this is very obvious when you are on antibiotics and many good bacteria gut flora dies, and then you may end up having a diarrhea. And I think nursing students probably have heard about C. diff infections. There is a lot of research going on in last 15 years or so, or 10 years, which shows the importance of the healthy gut bacteria. The the gut bacteria has been now linked to whether you are an obese or your body puts on weight very easily, or you're a skinny person. So weight gain, weight loss is associated with gut bacteria. Depression is also associated with gut bacteria. Some people do not have any genetics for having heart attacks, but the gut bacteria has been linked to those patients' heart attack. So it's a quite interesting area to do research on, or actually do a presentation, which is part of your assignments if somebody wants to do that. Okay. uh, Bacteria is actually not allowed to enter into the deeper tissue by what, because the epithelium cells are held together by tight junctions, and also we have lot of MALT, okay, which is your membrane associated lymphoid tissue is there. You also have a lot of T cells present, and then we also have lymphocytes, wandering lymphocytes. So there's a lot of IgA also present in there. So that helps us to protect against the bacteria. All right, so let's talk about the digestive process in the large intestine. So typically, the large intestine holds the material which has not been digested for 12 to 24 hours. There is no breakdown, as we mentioned earlier, except if there is something bacteria can actually digest. And the bacteria will also produce vitamin K and vitamin B12. Those vitamins, water, electrolytes, and salts are reabsorbed. So the major function is reabsorption of water, salts, vitamins, as well as propulsion of the feces to the anus or defecation. So as I mentioned earlier too, colon is not essential to live. Okay. So the motility in the large intestine is the contraction is the haustral contractions, or we also have gastrocolic reflex. So haustral contraction is the slow segmenting movements, and some haustra can sequentially contract uh, in response to when the food bulk comes into it. So for distension, the gastroiliac reflex is initiated by the presence of the food inside the stomach, and then this can activate 2 to 4 times a powerful peristaltic motions in which there is a mass movement of the colon. This is much more obvious in infants. If you have noticed in your younger children, infants, that when you feed them, they actually poop at the same time or just immediately after they uh, are fed. Some of the homeostatic imbalances of the large intestine or colon is diverticulus or herniation. This is when there is less fiber and people are constipated. So as the dried feces passes through, it actually injures the lining, and then that can cause pouches inside the wall of the colon. And you can have diverticula, which is herniation of the mucosa, and this is typically happens in the sigmoid colon, which affects almost half the people above the age of 70. You also have diverticulitis, means in which you have inflammation in the diverticula, and this may rupture or leak and into the peritoneal cavity, and that can become a life-threatening situation. Another one is the irritable bowel movement, or irritable bowel syndrome, which is IBS. This is a functional disorder of the GI tract in which you have recurring abdominal pain and stool changes, bloating, flatulence, nausea, depression, etc. Stress plays a very important role. IBS can be an autoimmune disease also. Crohn's disease is there, which is autoimmune. You can manage the symptoms of IBS with treatment plans. Then comes the last function of the intestine or the colon, which is called defecation. So the process of defecation means you remove or move the feces towards the rectum. And once the rectum has feces inside it, then that triggers that distension triggers the spinal defecation reflex. So once the feces move into the rectum, it stimulates the mechanoreceptors. Those receptors send the information to the spinal cord neurons, and then from there, the spinal reflex is initiated through the parasympathetic motor fibers, which will then help to contract the rectum as well as the sigmoidal colon. And then also under their action, you will have relaxation of the external and the internal anal sphincter. Okay, so the external is out in your control, skeletal muscle, but the internal is relaxed, and then external is also can be relaxed depending upon the convenience to defecate. Okay, and eventually the rectum will contract to expel all the feces out. So defecation can also be assisted by what we call as Valsalva maneuver. So in the Valsalva maneuver, what we does is we increase the intraperitoneal or abdominal pressure. So this can be done by closing the glottis, contraction of the diaphragm and the abdominal muscle walls. This increases the intra-abdominal pressure, and then the levator ani actually muscle contracts and it pulls the anal canal superiorly, which helps the feces to leave the body. Okay. Some of the homeostatic imbalances are there. Diarrhea is one of them. Watery stool is there. This can result when large intestine does not have enough time to absorb the water. And this could be because of the irritation of the bacteria present or the toxins produced by the bacteria present. And also if you move the digestive tract a lot, for example, running a marathon. Okay, so prolonged diarrhea is going to be very dangerous. It can result in dehydration and also into electrolyte imbalance, which can result into acidosis or loss of lot of potassium. Constipation is another one in which your diet does not have enough fibers. Okay, so it's very hard to pass that stool and it becomes more harder and harder. Okay, and that can be your improper bowel habits, lack of exercise, laxative abuses, etc. So I just put, just for the fun, the Bristol stool chart describes different types of stools the patients can or have been observed. Now let's look at the physiology of the digestion and the absorption. So digestion is a catabolic reaction. If you guys recall that catabolic reactions are degradative reactions, and they break larger macromolecules into small monomers, which can be utilized to regenerate the macromolecules of the body. For example, the carbohydrates can be broken down into sugars, simple sugars. Okay, so the enzymes are your amylases, sucrases, isomaltases, maltase, lactases, etc. Proteins can be broken down into amino acids by pepsins, trypsins, peptidases, etc. Fats and oils can be broken down into fatty acids and cholesterol by lipases, etc. Okay, so it's the enzymes which do the work. So the enzymes do hydrolytic function. Okay, so they use water and they break the water into two parts and then they add it to the chemical bond they are breaking. The second portion of digestive system is absorption. So the absorption process is that movement of the substances from the lumen of the intestine into the cells. So those cells are your epithelium cells. So they actually move from the lumen through the cell to the interstitial fluid and into the blood. Okay, so the substances are absorbed at the apical border, which is we also called it as brush border, and then they go through the cell and then they are released on the basolateral membrane, and from there they are taken up. Now the cells have lots and lots of tight junctions, so things do not just squeeze through this portion. Okay, so lipids can actually go through passively because they can go through the membrane. Polar molecules are required to have some kind of a carrier or a channel protein. Okay, so most of the nutrients are absorbed in the jejunum part of the small intestine before even it hits the ileum, or the chyme reaches the ileum. Okay, so let's look at the digestion of the carbohydrates. So our body cells can only absorb one simple sugar molecules. Okay, so those are the monosaccharides can be absorbed. So the typical monosaccharides our body absorbs is glucose, fructose, galactose. These are hexoses, means these are 6-carbon containing carbohydrates. Okay, and the enzymes which helps to digest carbohydrates is your salivary enzymes and also your pancreatic enzymes. So you have salivary amylases, pancreatic amylases, then you have brush border enzymes like dextrinase or glucoamylases, maltases, lactases, sucrases, which are disaccharides. They can break down disaccharides, sucrose, lactose, maltose, or polysaccharides, glycogen, and starch into smaller glucose molecules. So when starch is digested, the salivary amylase actually breaks it down into oligosaccharides, and the pH of the mouth is approximately 6.75 to 7. The pancreatic amylases do the function work on in the small intestine, and they break down whatever the salivary amylases couldn't break, and they produce small chunks of the sugars that are known as oligosaccharides. And then the brush border cells, the brush border edge has enzymes like dextrinases, glucoamylases, lactases, maltases, and sucrases, which will change these oligosaccharides into the monosaccharides. Okay. So here is a picture which explains it. So what happens is in the salivary amylase, this is in the mouth, the glucose and galactose can be absorbed via a co-transporter called sodium glut. Fructose can pass through the facilitated diffusion, and all other monosaccharides, they leave the epithelial cells via facilitated diffusion and enter the blood. So pancreatic amylase is in the small intestine. It will break down the starch and the disaccharides into oligosaccharides and disaccharides, and from there it goes lactose, maltose, and sucrose. And then lactose is made up of galactose and glucose. Maltose is made up of 2 glucose molecules, and sucrose has 1 glucose and 1 fructose. Okay, so they are present in the brush border of the small intestine. These enzymes are present there. How is the carbohydrates absorbed? So glucose and galactose, they can be actually absorbed by secondary active transport, or what we call as co-transport systems. Okay, so it is the co-transporters which actually transport the sugars most of the time. Okay, and they are always transported with the sodium ions. Okay, so then they enter from the brush border side, and then monosaccharides are released, and then at the basolateral side these monosaccharides are then transported through the facilitated diffusion, because the concentration of them is more inside the cell than in the blood, so they go from there. Okay, sodium is also taken out of the cell by sodium potassium ATPase, which actually is involved in maintaining the resting membrane potential. Fructose is also actually entered into the cell through diffusion, and then it also exits the cell through the facilitated diffusion. All right, so let's discuss now how are the proteins digested in our body. So first question is, where does the proteins come from in our body or in digestive system? Is through the food. So that is the dietary protein, whatever we intake. Other thing is the digestive enzymes which are released inside our digestive tract. That's there. Mucosal cells also release some proteins, or the cells which are actually being degraded. That's also there. All of them are digested to amino acids by enzymes called proteases. The protein digestion actually starts in the stomach. So in the stomach, it's the HCl starts breaking it down. Okay. And the enzyme is the pepsin. So the stomach acid changes the pepsinogen into pepsin, and then pepsin keeps activating more pepsinogen into pepsin, which denatures the protein. And as this chyme in which you have oligopeptides, means little longer amino acids chains, they passes into the duodenum, and then this pepsin becomes inactive because in the duodenum we have the pancreatic juices as well as the bile acids join, and the pH goes very, very high. Now over there, the pH has gone up from pH 2 to pH 6, and this place we have the pancreatic enzymes, trypsin, chymotrypsin, elastase, carboxypeptidase, etc. So those enzymes works on these oligopeptides and makes the amino acids. And then in the small intestine, we have another other enzymes which are present on the brush border side of the cells which are lining the lumen of the small intestine. These are aminopeptidases or carboxypeptidases or dipeptidases, and ultimately we create is single amino acids, and then amino acids are absorbed by the blood or surrounding material there. This picture shows the role of different types of enzymes, where they are located for the proteins. So in the stomach, it's the pepsin, which is going to act on the proteins and make them into large polypeptides. And then as it moves to the duodenum, and then in the small intestine, it's the pancreatic enzymes, which is trypsin, chymotrypsin, and carboxypeptidases, makes it into small polypeptides or small peptides. And then in the intestine, the brush border enzymes, which are aminopeptidases, carboxypeptidases, and dipeptidases, will convert all these small polypeptides or small peptides into amino acids. Or some dipeptides or tripeptides can be there, and then they can be taken up by the capillary blood into the villi and then transported to the liver via the hepatic portal vein. So absorption is done by several different ways. So they have different types of carrier proteins, and they are mostly coupled with active transport of sodium. So sodium and amino acid transporters will be there. Dipeptides and tripeptides are absorbed actively by proton dependent co-transporters, and then they are digested to the amino acids, and then the amino acids at the brush border size will transport them out through the facilitated diffusion, and then they are taken up by the blood. Digestion of the lipids. So lipids are pretty large molecules, so they require pre-treatments. So they require emulsification by the bile salts. Okay. But this bile salts just emulsifies them, makes the smaller pieces of them. It doesn't chemically digest them, so there is no bond breakage occurring. So it starts in the mouth with the what we call as the lingual lipase. So the lingual lipase can break down it a little bit. Okay, and then it goes to the stomach. In the stomach, you just have the mixing, and there is very little gastric lipase. So there is little digestion of the lipids in the stomach. But when it moves into the duodenum, that's where it comes in contact with the pancreatic lipases. That's where we break down the lipids. The neutral lipids are changed into fatty acids and monoglycerides. And then the fatty acids, the glycerol and the short chain fatty acids, are then absorbed by the capillary bloods in the villi of the small intestine, and then they are actually taken up by the hepatic portal veins, and then goes back to the liver. Here's the fat and the lipid digestion. So this is unemulsified triglycerides, which is in your mouth. It is the lingual lipase, extremely small amount effect. Then in the stomach, the gastric lipase also have very little effect, but it is only in the small intestine. Emulsification occurs, that means breaking down into small parts by bile salts, as well as then we have the pancreatic lipases, which will then break down into glycerol and fatty acids or monoglycerides, which were then taken up by the they're taken up by the lacteals, etc., and eventually into the hepatic portal vein. So the absorption of the lipids we have talked about earlier too. So this is done as monoglycerides or fatty acids. So the bile salts and the lecithin, they actually coat the fat globules, and they make these droplets onto which the enzymes can occur. And then from enzymes, once they occur them, then they form the micelles, etc., which can take them into the intestinal epithelium, see where actually they can combine with other packing material like proteins, etc., which carry them, and that's what forms is called chylomicrons. Okay, so once you have the proteins coating them is the chylomicrons, and then they diffuse into the interstitial underlying interstitial tissue, which is typically areolar connective tissue, in which we have the blood vessels and as well as we have the lymph vessels. So they enter into the lacteals because of their large size, and the lymph vessel have fenestrated vessel walls, and then it is taken up by the portal blood system, hepatic portal blood system, for distribution to the liver. All right, so now let's look at what happens after the body absorbs the amino acids or the fats. Remember, sugars can be utilized as such by cells to generate energy. But what happens to the amino acids? So recall, an amino acid has a carboxylic group, COOH, and central carbon is there, and then an amine group is there, NH2 group here. I am going to try to make it as a formula. So you can remove this amine group, and what is left is an acid group. Okay, so each amino acid has its corresponding keto acid. Okay, if you give amine group to that keto acid, it becomes the amino acid. So that reaction in which amine group is actually transferred to a keto acid is known as transamination. Means we are transferring an amine group. Okay. Say for example, we started with an alpha ketoglutaric acid and we use any amino acid, say glycine or any other one. That will be changed into its keto acid, and then we will have glutamic acid. That's how the body actually cells make the new amino acids. Okay. They switch the amine group from one amino acid to a keto acid. You can also remove these amine groups, and the second way you can remove this amine group is by oxidative deamination, means the ammonia is released, and then ammonia in the liver combines with CO2, carbon dioxide, to give you urea, and then urea is taken to the kidneys, and then it is excreted as urine. Okay, the keto acids which are produced, which are the after the removal of the amine group, then can actually modify them and then they can enter the blood, and then they can enter into the cells, and they enter into what is known as Krebs cycle, which we will discuss in next chapter. Lipids, same way. Lipids are made up of glycerol, and glycerol then can enter into what we call as glycolysis. The sugars are broken down by glycolysis, and the fatty acids which are released, they can be converted into acetyl-CoA, which is through a process called beta oxidation. Okay, so that through beta oxidation, the acetyl-CoA then all enters into the Krebs cycle. Okay, so Krebs cycle has many things which can enter into it, and we'll discuss this in next chapter, which is nutrition and metabolism. Now, all right, so the last macromolecule which is left is the nucleic acids. Remember, nucleic acids are made up of nucleotides, which is a nucleoside plus a phosphate. And what is a nucleoside? A nucleoside has a sugar and a base, nitrogen bases, okay, are present. So there are a couple different types of nucleic acids, two major types. One is RNA, the other is DNA. So the enzymes which breaks them down will be ribonucleases and as well as deoxyribonucleases, and they will convert them into their nucleotides. From the nucleotides, we can use the enzymes, brush border enzymes present in the intestine cells, which will change it into nucleosides, and the enzymes which work on there is nucleosidases, changes it into sugars and nitrogen bases. The sugar is pentose, and then phosphate is released. Pentose can actually enter into glycolysis through another pathway is called pentose phosphate pathway. And then they can be absorbed. Sugars can be absorbed by the epithelium in the intestine and then can enter into the blood. So here we have in a more picture form, so nucleic acid by pancreatic ribonucleases and deoxyribonucleic acids. They'll break down into pentose sugars and nitrogen bases and phosphate ions. They can be absorbed by the capillaries of the blood in the enzymes in the villi, and then can be converted or transported into hepatic portal system, and then goes to the liver or the cells. How are the vitamins absorbed? So vitamins typically are absorbed in the small intestine. The fat soluble vitamins like A, D, E, and K, they can be carried by micelles, and then they can diffuse into the absorptive cells. Most of the ions like iron, calcium, magnesium, and zinc, etc., are absorbed in the duodenum. Okay. The jejunum absorbs your glucose, vitamin C, B12, all the vitamin Bs, etc., pyridoxine, folic acid, etc. They're all done in there. Water soluble vitamins like C and B are absorbed by diffusion or by passive or active transports. In the large intestine colon is where we have vitamin K and vitamin B absorbed from what is generated by the bacteria present inside the large colon. How are the electrolytes absorbed? The small intestine is where most of the ions are picked up by the active transport. Duodenum is where we have iron and magnesium and calcium taken up. Sodium is also coupled with active absorption of glucose and amino acids, and chloride is also transported actively. Potassium can actually diffuse in response to the osmotic gradients, and it can be lost if there is poor water absorption. Okay. Iron and calcium is absorbed as we need it, and iron is stored in the mucosal cells is with ferritin. A protein ferritin binds to it, and then it is transported in the blood by another protein called transferritin. So it's good to remember these names. Calcium is absorbed from the intestine under the action of parathyroid hormone, or what we call as PTH. How is the water absorbed? Actually, uh, approximately 9 liters of water, which is mostly from the GI tract secretions that enters into the small intestine. 95% of it is absorbed by osmosis because the salt concentration around is more, and the rest is absorbed in the large intestine. Okay, so the net osmosis will occur if the concentration gradient is established by the active transport of the solutes. Okay, uh, so water uptake is coupled with that of the solute uptake, which is your sodium. So for example, you can drink around some water, 1200 ml or 800 grams with the food. So the water comes in 1500 ml of salivary secretion per liter, approximately 2 liters of gastric secretions. Then we have 1.5 liters of pancreatic secretion, half a liter of liver secretion, and then they all go through this small intestine, jejunum and ileum, so where we approximately absorb 900 close to 9 liters, and then only 100 liters and is released, 100 ml is released along with the stools. Very little is left in the stools. One of the homeostatic imbalance which can occur in the absorption is malabsorption of nutrients, and then there can be many different causes for it. It could be anything that interferes with the delivery of, say, bile salt or pancreatic juices, etc., or your mucosal is damaged because of the bacterial infection or some antibiotics. Or you may also have what we call as gluten sensitive enteropathies, or what is known as celiac disease. So this is in which the gluten, which is typically present in many grains, your body reacts to it and starts producing antibodies to it, and it can cause the immune system cells damage the intestinal villi and the brush border of the intestine. And you can manage it by eliminating all the gluten from the diet. All grains except for your rice and corn has gluten in it, but these days it's much easier to get gluten-free food.